Hello everyone, welcome to biology lecture. Myself Somnath Palicha here. We are going to start with our today's new chapter that is called as a transport in plants. Okay, class 11 NCRT topic. So let's talk about this transport in plants. For this chapter to understand, we had completed a very important chapter, you can say, that is the anatomy in flowering plants, right? Or you can say anatomy of hair plants. So we had already discussed about their xylem and phloem, all those conducting elements. Okay, the same thing will be coming over here. Along with this, we had also having a concept with respect to the, you can say, active and the passive transport. And even here we are going to deal with the examples called as a osmosis, diffusion, facilitated diffusion, and many more things. Okay, because all this procedure will be going to help us to understand how the transport actually takes place in the plants. So let's move ahead. I'm starting with the, you can say, NCRT line of the chapter that is called as a in plants substances are transported over a short distances as well as long distances. This is very clear cut statement है ना. So as you can see, in plants small distances can help the plant to diffuse the things very easily from one cell to the another cell because having the less intercellular spaces and all. Okay, and due to this a small distance can be covered, and for the long distance we need a vascular bundles over here. Okay, now this short distance transport, whatever things are happening over here, you very well know there is a cytoplasmic streamings are available, right? So it is mainly done by the cytoplasmic streaming, which is you can say helps in the diffusion process, and the things will be done for you. Now see what is written over here. Short distance transport is mainly by the cytoplasmic streaming and diffusion supplemented by the active transport. Now transport over a long distance we need a vascular bundles, and it is also called as a translocation. So process is called as a translocation over here. Very important statement. As you can see, it is a kind of question also, a very basic kind of question from here. Let's talk about what is active transport and the passive transport. Everyone must be familiar with this because we had already deal with the cell unit of life chapter also. There also we had having a talk on this. So now the same thing. Let's see over here. Yes, ATP is required. In passive process, ATP is not at all required. Yes. Okay. So can takes place against the concentration gradient, but here it takes place through the concentration gradient, that is higher concentration to the lower concentration till it reaches to the equilibrium. Now it is a very sensitive to the temperature. If suppose it is absent, or you can say not temperature absent, you can say more or less temperature and all those things, inhibitor like cyanide and all, if it is present, then the obviously the process is going to get affected. Okay, but in case of the passive transport it is not like this. There is no factor like temperature and inhibitors going to affect the process. Now example which come for the here active and the passive transport is that, see, for the active, sodium pump, potassium pump, calcium pump, and endocytosis and exocytosis activities are the examples over here. Whereas if you take example for the passive process, that is the osmosis, diffusion, and facilitated diffusion is the very great example which is directly asked in the examination. So let's try to understand this only. Lekin, before entering into the detailed part of the chapter, you should keep one thing in the mind, that note for you. Okay, see, osmosis, diffusion, facilitated diffusion, application of the osmosis after affecting the factors affecting the rate of diffusion, all these things you have to study from the lecture video which is called as a cell the unit of life. Okay, cell the unit of life part 2 lecture video has been uploaded on your YouTube channel. From there you can study. I will be just providing you the link in the description box of this second part of the cell the unit of life chapter. You have to go through that before you should like go through rest all the part of this lecture. Okay, understood very clearly what I said? You need to go through first the cell the unit of life part 2 lecture before entering in this topic. So now let's talk about water potential. So from here actually the transport of plants, transport of transport topic which we are going to deal over here, that is started already. See, energy purchases by the water molecule, that is nothing but called as a water potential. Greater the concentration of the water, greater will be the kinetic energy. Also keep in mind, okay? Or you can say water potential. Because of the liquid, the gaseous molecule are always in a random motion. Now here you should keep one thing in the mind. Whenever the water molecule, if we take as an example, or gaseous molecule will take as an example, they will be always in a random motion. Okay, that random motion will give a kind of kinetic energy to the molecules. Okay, so that molecular energy, you can say over there, kinetic energy, that is actually called as a water potential. Now for note, you should keep always one thing in the mind. whatever the note will be coming on the screen, these are all direct questions. Okay, so see, pure water has the maximum or greatest water potential. What is pure water? Means there is a negligible amount of the impurities, no impurities at all, and that is considered as a zero. So zero is what? So maximum water potential of whom? Of pure water. I'm repeating again, pure water has the maximum water potential and that is considered as a zero. Zero is here means negligible amount of the impurities. Now, movement of the water will takes place from the system with the higher water potential to the system of lower water potential. Suppose two kind of the liquid I kept in a contact which each other, then you should understand that things will be moving from the higher water potential to the lower water potential. Higher means from the zero, okay, you can say, and lower means there is an impurity present in that particular water. So let's move ahead. Always water potential is denoted by the psi and the unit is Pascal. Okay, water potential of the root here, which is almost equal to the -1 to -2 for any kind of the solutions. Okay, so water pressure, I had just one of the example given to you over here. Water potential for the root here is almost -1 to -2 for any solution. What this minus sign, minus values indicates over here, that much amount of the impurities present over there. Okay, you can say 1%, 2% impurities can be seen in case of the root hairs. Okay, water potential. So let's talk about here factors which can affect the water potential. Okay, so I'm talking about here which are the factors which are responsible for water potential to get affect. So जैसे कि one is called as a solute. Okay, so solute potential word has been came on the screen. When the solute potential increases, water potential decreases. What do you mean by the solute? Take as an example salt plus water. So more solute will be getting added, so water potential is going to decrease because the pure water molecule are going to decrease. Are you understanding? Okay, in that sense. So pressure. So again the term will be coming on the screen, pressure potential. Pressure potential is always considered as a positive. Okay, when the pressure potential increases, also the water potential is going to increase. Keep these statements in your mind very clearly because this can be a very good question from this chapter. Okay, next one more is the gravity. So gravity potential, but gravity potential is negligible effect up to, you can say, 5 meter in the height. Okay, from the ground level to the 5 meter of the height, it shows a negligible effect. Okay, initial equation, if you see, if you write with respect to the all these three above mentioned points, so this is what the equation you will be getting. Psi W, that means water potential is equal to the solute potential plus pressure potential plus gravity potential. But you know that gravity potential is negligible up to 5 meters. Therefore, by skipping that particular point, you will be getting a very good equation on the screen, that is the pressure, say water potential is equal to solute potential plus pressure potential. So this is what the equation you should keep in your mind. Okay, from this equation, then the many questions can be come on the screen. Okay, now note, pressure potential can be negative if, see, when the transpiration has been taken place. When we consider the pressure potential can be negative because it is working against the gravity. Okay, things has to move from the ground level to the upper level. Okay, that is possible due to the transpiration, that is the opening of the stomata. Suction pressure will be created, a kind of transpiration pull which will be taking the sap towards the upper side. Okay, so through this you should understand that pressure potential is over here negative. Clear hai? So let's move ahead with respect to the osmotic pressure. This is a very next topic which we are discussing right now. The pressure which is required to stop the osmosis, that is the movement of the water completely, is known as the osmotic pressure. It is always given by the pi. Okay, keep this again in your mind. Very, very important. Also known as solute potential. Now see, osmotic pressure pi and osmotic potential psi s, okay, are numerically equal, but pi is always negative to the solute potential. Okay, I will explain you this, try to understand. See, always pi value will be considered as a positive and the solute potential or osmotic potential value will be considered as a negative. Therefore the equation can be written as pi is equal to the negative of the solute potential, or you can say osmotic potential. Understood from here? But numerically they are equal. Let's move ahead. Diffusion pressure deficient. Again, very, very important concept over here from your NCERT textbook itself. Okay, so see, it is defined as the amount by which the diffusion pressure of a solution is less than its pure solvent. Okay, क्या बोल रहा है? Amount by which the diffusion pressure of a solution is less. Diffusion pressure of the solution जो होता है, that is considered as a less as compared to the pure solvent. इसको समझना बहुत आसान है. As you can see on the screen, there is one example given to you. There is a turgid cell and as well as there is a flaccid cell. Both are placed together. Okay. Now what will happen? See, osmotic pressure for the turgid cell has been given you the 10. Okay, and the turgor pressure given you the value as called as a 6. Okay, and what is the formula for the calculating the dpd? That is a op minus tp. Okay, that is tp. So op minus, if I write wp also, then also no problem, because you know that turgor pressure is always equal to the wall pressure. Okay, whatever the amount of the wall pressure will be there, that is equal to the turgor pressure. Okay, now op minus tp is there, so just substitute the value 10 minus 6, you'll be getting answer as a 4. Okay, so this is the dpd for the turgid cell. Now let's talk about the flaccid cell. Suppose there is a osmotic pressure over here is 10. Okay, and the turgor pressure over here is 4. So calculate the dpd over here, you will be getting your answer as a 6. So this is high, right? So see, water moves from the lower dpd to the higher dpd. It is very different, right? It is moving from turgid cell to the flaccid cell. Understood very clearly over here, because turgid cell has the dpd 4, that is the less, as compared to the flaccid cell dpd is 6. Okay, this is how you just easy way to understand the movement of the substances, okay, from the lower dpd to the higher dpd. Now we are entering into the very interesting topic which you already familiar with, imbibition. Okay, so let's see. Imbibition has to take place. There is always a absorbent substance is required. Take example dry seed. Dry seed is one of the very good example. Whenever you need to like cook the dry seeds before one night, okay, you will be keeping this thing before one day, you will be keeping this thing seeds in your water, okay, in a water vessels for soaking the seeds, right? So soaking seeds means what? The water will be getting entering into the seed. You need to see there a lot of changes are happening, increase in the volume, okay, and pressure will be developed inside the seed and all those things. So these are all general whatever scenario you are looking in your life, that is all we are going to discuss over here. So now try to recollect this thing. See, imbibition pressure. First point is coming on the screen, that is the imbibition pressure. That is a swollen cell produces a large pressure. Always keep in mind, swollen cell produces a large pressure. For example, one dry seed having the 1000 atmospheric pressure. You can take as a pea seed also as an example to understand this. Okay, 1000 atmospheric pressure has been developed by the one dry seed. Here the kinetic energy of the water is released. See what is written over here. Here the kinetic energy of the water is released as heat energy, okay, and called as a heat wetting. So what is the term given to this kind of energy which is released in the form of heat? We call this as a heat of wetting, or another term we can use for this, that is a heat of hydration. Okay, one thing also you able to see that volume has been increased, है ना, because of the entry of the water. Now dry seed always will be having the negative water potential. Always keep in mind, dry seed will be having the negative water potential. Therefore the things has been moved from the higher water potential to the lower water potential, right? Imbibition stops as soon as the equilibrium is reached. What do you mean by equilibrium? Means everything has been balanced. Now no net movement will be seen over there. Okay, let's move over here. We are entering into the very interesting topic again, called as a wilting. So what is wilting? Again, a general life experiment you can see takes place due to the availability of the water is less and evaporation and the transpiration is more. Means whenever you will see the plant, okay, and the plant is like in a condition where there is a lot of sunlight, okay, heat energy is there. Means actually you can say the temperature is more. Just keep the plant in that particular situation. You will see that wilting is happening because transpiration is there. Water will be evaporated as a water vapor. Okay, so more number of the water will be getting lost. So what will happening over here, there is a water stress condition will be developed, okay, and plant will start wilting. So wilting topic has been started already, so we need to understand the types of wilting also. So just focus over here. There is an incipient wilting, that means symptoms will not be seen from the outer side, but the plant is actually in a stress condition. Okay, temporary wilting, that means what, you just need to pour the water. By looking at the plant itself, we will come to know there is a plant wilting has been process is happening over there, means plant is already in a stress condition, means there is no water for the plant to take. Okay, now see, symptoms can be seen from the outer side. For example, leaf curls, means leaf will be started curling, okay, it will be started falling down. That kind of scenarios you will able to see on the plants. Okay, next, permanent wilting. symptoms can be seen again from the outer side, but here if you add the water to the soil, again the plant is not going to use the water because the plant root system has also been died over here. Okay, so that is what the scenario. Three types of the wiltings you had studied: incipient wilting, temporary wilting, and the permanent wilting. Again, you can say one of the very important topic for your chapter. Next, movement of the water. This is a very very important part which we are dealing right now. Movement of the water. So apoplast movement. Water does not cross a membrane, so it moves from the cell wall. Maximum volume will be takes place over here. So maximum volume will be transported according to the apoplast movement, and sim plus less amount of the movement will be occurring of the water. How it taking place, that we are going to discuss from the white screen. Okay, so here there is a white screen present on in front of you. So just focus on this. Now, suppose this white screen I am drawing something for you people. So let's talk about this is the root hairs. Okay, and this is the epidermis. Fine. So these are an epidermis, and this will be called as a root hair. I will be labeling soon this also. So see over here. Fine. This is the root hair. Now, if I draw the cells over here inside, see, before drawing the cells, let's draw one more thing that is called as the endodermis. Okay. So these are the endodermis regions. This is the endodermis. So let me label this also. Endodermis. Now this endodermis has a two kinds of the cells. One which is called as a passage cell. Okay, and another will be there that will be called as a, you can say, Casparian strips will be present. Okay, what I said, passage cells and the Capsarian strips will be present over there. Now we need to understand this thing. See, let's change the color over here now. Okay, so let's try with the brown. So I will be drawing something in the endodermis region. Try to recollect that part. Okay, so suppose this cell over here has a one particular Casparian strip. Okay, see, here there is a Casparian strip. Let's draw here also there is a Casparian strip. Okay, and let's draw here also. So some cells will be having the Capsarian strips, as you can see. This is a labeling over here, and some cells will not be showing that Capsarian strips, so they will be called as a passage cells. Okay, so this is very clear to you. Now, in between this, there is a presence of cells which are normally called as the cortex cells. है ना? So let's draw the cortex cells now. So suppose these are my cortex cells. Okay, these are normally parenchymatous cells, loosely arranged cells, you can say, having intercellular spaces. Okay, living cells, so many things you can just easily define, है ना? So from here let me draw some also here. So here there is a passage cells, sorry, the cortex cells. So let me take this also labeling over here. So these are cortex cells. Fine. So now let's move ahead with the one more criteria that is called as the vascular bundles. So let me draw over here. Suppose this is xylem and suppose this is a phloem. So I'm writing over here, this is a phloem and this is a xylem. These are vascular bundles. In between also there is a parenchyma cells. I'm not interested in that. Okay, let's for your understanding purpose draw that also. Chalo, bana lete hain. Okay, so some cells I had drawn over here. Okay, so my diagram is ready for you. Now let's talk about first apoplast movement to take place. Okay, which moment? Apoplast movement we are going to discuss over here. Now from the root here, the water molecule will be entering inside, right? Apoplast I'm explaining over here. Water molecule has been moved over here. Okay, now water as soon as it has been absorbed by the root hairs, the things will be started moving from the cell wall. Understood? It is not entering into the cell. Try to understand this point very clearly. So things has been moving from the intercellular spaces or through the cell walls of the, okay, cells over here. So see, okay, and now it has been reached to a particular region towards the endodermis, but it's tried to go inside, but it is not possible because there is a Capsarian strips. And what is this function of the Casparian strips is that that is impermeable to the water, means they will not allow the water to move inside the endodermis region. Okay, so what will happen to this? They will try to go from the passage cell. As soon as the passage cell has been crossed, okay, so it will be entering into the cell wall to cell wall, and it will go to the xylem. Okay, and then the conduction will be taking place. This is what the apoplast movement has been explained over here. So what is this kind of movement called as a apoplast movement? Understood? Maximum conduction will be happening in this way only, apoplast movement through the cell wall to cell wall and the intercellular spaces. Now let's talk about here with respect to the symplast movement. But you should understand, before entering into the symplast movement, you see that apoplast is not able to enter through the these capsarian strips, right? At this area the things are not moving inside, but things can pass from the passage cell. So only for this much interval of time, the apoplast movement has to convert into the symplast movement. Understood? So here there was a symplast movement. This is here to understood, very very important term over here. Okay, क्लियर है? सो सिम्प्लास्ट एपोप्लास्ट कितना भी कर ले उसको एंडोडर्मिस के थ्रू सिम्प्लास्ट ही करना होता है. Okay, that is a very very important thing which you should keep in mind. Now let's talk about here with respect to the symplast movement. Okay, so same water molecule over here. Okay, a water molecule with the black color, focus on this, taken up by the root hairs. Okay, from the root hairs things will be enter into the cell, from the cell to the another cell, and from another cell to the another cell. Like this way it will be keep on moving. Okay, and it will enter into the passage cells through this, and the things will be enter into the xylem finally. Okay, so what is this? Again a symplast movement. Okay, so this was what? So this was a symplast movement. So symplast will not be facing any kind of problem. It will be doing through epidermis or through the endodermis. It will be showing from start to end. It will be like symplast movement itself, but less amount of the water will be transferred in such way. Okay, but again, if suppose water is trying to go from here, capsarian strips will be coming into the endodermis region. Okay, the water will not be able to enter inside the you can say endodermis region. Okay, means not be able to pass because the capsarian strips are present over there. These are you can say impermeable to the water. So I hope the concept is very clear to you all. Okay, let's move ahead with the next part. So I'm entering into the PDF again, so that we can continue with the lecture. See, note. So this is a note which you have to keep in mind. Endodermis shows only the simplest movement due to the some cells of the endodermis which shows the capsarian strips present over there. Understood why the simplest movement has to take place in the apoplast region? Okay, because of the capsarian strips present over there. Okay, it is a kind of suberin deposition. Because of that, it is showing the impermeable to the water. Now we are entering into the ascent of sap topic, that is nothing but water translocation. Okay, it is very simple. Only thing is that two theories which we have to deal over here. First is called as the root pressure theory. Okay, so what is that? Root pressure terms given by the called as a Stephen Hales. So Stephen Hales was the scientist who has talked about the root pressure. Okay, now there was another scientist whose name is Stocking. Okay, Stocking. Pressure development in the tracheary elements of the xylem is due to the metabolic activities. Is what? Is due to the metabolic activities of the root called as a root pressure. Okay, so note what is the note says over here? Water from the soil is absorbed by the root hairs. This everyone knows, right? Water from the soil is absorbed by the root hairs. If a plant is transversely cut above the soil surfaces, sap from the xylem oozes out due to the root pressure. Now, suppose I draw over here a kind of a plant. Okay, a kind of plant, and suppose here there is a ground level. Okay, let me go to the white screen only, that will be better for you to understand the concept. Now suppose there is a plant. Okay, here there is a plant and this is a ground level, like almost 1 meter you can say. Okay, at 1 meter of height or just little bit above from the ground surface, you had given a cut to the plant. Okay, when you given a cut to the plant, this is how it will be looking, right? Smaller size, है ना? So once you start looking at this particular region, okay, once you start looking at this particular region, you will able to share, you will able to see there is a water droplets. Okay, on the cut portion you will able to see there is a water droplets coming over there. Because of this, see what is happening over here? Root pressure. Okay, some kind of water as like see when the capillary actions takes place, there is a chance of water to get uh, raise particular height. Okay, is started going to raising a particular height, so that will be like around 1 meter. Okay, that is a minimum height you can say. Till that height the water will be started moving upward. Okay, but since you had chopped the particular plant at 1 meter of height, you will see that still there is a action of capillarity. Okay, means what, the water droplets which is coming out from the xylem part. Okay, and that part will be visible for you people. Okay, so that is what the action of capillarity you will see over here. So let's try to go with the PDF now. Okay, so understood this much. Now see what is the very next point over here. Water potential of a soil is closer to the zero. That means what, water potential, water which is present into the soil is like almost you can say good amount of the water potential it has, and the higher as compared to the root hairs, but it is very higher as compared to the root hair part. Means root hair somewhat it can be the solute concentration will be more, right? So things you know that how it will be moving from the higher concentration to the lower concentration, that is from the soil to the root hairs, right? Therefore the water will be enter into the root hair from the higher water potential to the lower water potential. Understood very clearly. Now let's try to go ahead. See, root cortex cells absorbs iron actively. This is a very important statement. Root cortex cell absorbs ions actively. That means it is a ATP expenditure, okay, from the surrounding cells, result in the lowering the water potential. Now this results in the movement of the water from the root hair to the cortex cells. Means the things has to move from the root hair to the cortex cells. Okay, so cortical cells has a lower water potential, therefore the movement has been taken place from the root hair. And understood this concept very clearly. So you should try to understand every time where the concentration means where the water potential is increasing and the where the water potential is decreased. Okay, and how the movement has been taken place. Actually it is from the higher water potential to the lower water potential. This is the basic thing which you should carry on the concept over here. Now see, now the water will enters into the xylem and ions will be actively absorbed, and the movement of the water is a passive, that is due to the osmosis process from the higher concentration to the lower concentration. What is happening, actually movement of the solvent has been taken place, right? And that is the water over here. Okay, so these two lines are very important for you. Ions are actively absorbed and the movement of the water is done by the passively process called as a osmosis. Now root pressure increases when the water absorption increases. Okay, when the root pressure increases, water absorption is also going to increase. That is due to the less transpiration also. Okay, in summer it is very lower. In summer it is very lower. Keep this in mind, because things has to be stopped, otherwise continuously transpiration will be taking place and all the water will be getting evaporated. That is a basic concept you know very well. Okay, if the root pressure is more and the loss of water is less, then the liquid water ooze out through the special openings of the veins near the margin of the leaves called as a hydatodes, and the process will be called as a guttation. Suppose if I draw a leaf over here, okay, here there is a leaf, so at the margin of the leaves, okay, there is a presence of the small tiny openings, and those tiny openings are taking out the excess of the water, okay, in the form of water droplets. So normally when you go for the jogging purpose in the early morning, like in the winter season, okay, like if you do a run on a particular herbaceous plants and all, you will see there is a formation of water droplets on that. Okay, so while running you can feel the water droplets. If you see the shoes, they will be getting wet because of these water droplets and all those things. Okay, very common thing which you had noticed in your life, but the process will be called as a guttation, and the water which comes out from the leaf margin area, from the leaf margin openings, okay, these are called as a hydatodes. Okay, so these are the two very important concept over here, extremely important you can say. They can ask for the examination point of view. Okay, now second theory is that physical theory, which is of three types. So number one is a capillary, okay, capillarity. So it is given by the scientist called as a Boehm. What do you keep in your mind? That see, water can rise up to a 1 meter in the capillary, that is 0.03 mm diameter. If the capillary shows this much thickness, 0.03 mm, it's not that thick, right? It is a very fine thickness. Okay, very fine lumen will be there, and that lumen is enough. Like you can say, whenever you want to have a fruity, okay, and if you are using a fine straw, the fruity will come very suddenly. Okay, and when you take the fruity and if you have a very thick straw, it will take little time. Okay, because water increase will be like very less as compared to the small capillary, because the lumen is very small over there. Diameter of the lumen is very small over there. Okay, and the diameter of the lumen, if suppose wide, it will not be showing such effect over there. Okay, so this is the two things which you have to keep in your mind very clearly. Now We have the cohesive and adhesive force theory also. So let's talk about this. Cohesive force means what? It is the attraction between the same type of the particles or molecule, that is between the water to water. I will be explaining you this on the white screen. And adhesive force means always different molecules attractions will be there. Okay, so water and the lignin molecule of the xylem vessel. To understand this better way, let's go on the white screen. Okay, so I will be taking you over here on the white screen. Try to understand the concept over here. Adhesive force and the cohesive force. Suppose it is a xylem vessel now. Okay, what is this? This is a xylem vessel. So xylem vessels will be having some kind of lignin depositions. Okay, so lignin molecule will be there. So these are the lignin molecule. Okay, let's draw some water molecule also. So I'm drawing here some kind of water molecules also. Okay, so suppose these are the water molecules. I'm talking here about the xylem element. Okay, so vessel, xylem vessel. So suppose this is a water molecule has been prepared over here. Now what will happen? First, let's talk about the adhesive force. Okay, that is the between the different molecules. So water molecule will be showing the attraction between this uh, first water molecule. See, first layer of the water molecule will be showing the attraction between the lignin molecules. Understood? Because it is a lignin also, no? It is a made up of some lignin like substance. Okay, that is also having some kind of molecule. So this is the attraction between these two. So this water molecule will try to move upward because of the attraction. Are you understanding? So they will be trying to move upward, but as soon as the first layer of the water will start moving upward direction, now there is a cohesive force. Cohesive force between whom? Between the water to water molecule, means same molecules. Are you understanding? So this first molecule will move upside, so but obviously it will going to take the second molecule with him, and the second molecule will bring the third molecule with him, and this is how actually the rise of the water is going to take place. Understood very clearly? So what is happening over here? Cohesive and the adhesive force are working. Adhesive force that is taking place between the water molecule and the lignin substance, or you can say lignin molecule of the xylem, you can say xylem vessels. And cohesive force has been taken place between the water to water molecule. So first water molecule will be always showing attraction between the lignin molecule, and it will start moving upward. But soon, because of this process, the water to water molecule will be started pulling the water molecules to the same height. Okay, this is a basic concept over here which you had understood very clearly. Now this can be understood by one more example. Suppose there is an engine. Okay, and here there is a compartment, first compartment. Okay, so it is a compartment and here it is an engine. So engine and compartment, there is a link. Okay, engine and compartment shows there is a link. So as soon as the engine will start moving, this compartment is also going to start moving, right? But this compartment is actually attached to the some other compartments. Are you understanding? So as soon as the first compartment, which shows attachment to the engine, when engine start moving, this first molecule means first compartment will also start moving. So but the first compartment actually attached to the same type of compartment, so it will also start moving forward direction. Understood? This is the basic thing which you should understand from here. Okay, so cohesive and adhesive force theory also plays a very important role to understand the concept over here. Okay, this is how the transpiration actually taking place. And see, now we are entering into the very next part, that is, this is only known as a cohesion adhesion theory, which I already told you, and the next third one is called as a transpiration pull theory. Okay, first was the capillarity, second was the cohesion and adhesion theory, now there is a transpiration pull theory. So this is happening because of the stomata. When the stomata in the daytime open up, okay, water vapors and all those started moving out, suction pressure will be developed. Okay, that suction pressure will work on the, you can say, xylem vessels. Okay, and it will start taking the ascent of the sap, will actually the process is going to be taking place over here. So see, before entering into the explanatory part, you should understand that transpiration pulls develops a negative pressure because things has to move against the gravity, right? And that pressure is approximately minus 77 to minus 142.9 bars. Okay, this is the value given to you. It is measured by the pressure bomb technique. Okay, it is given by the pressure, it is actually measured by the pressure bomb technique. Now this negative pressure helps to pull the water column up. Okay, understood? So negative pressure helps here to move the water column into the vessels, and it will move towards the upper side. So loss of water in the form of water vapor through the stomata is known as the transpiration process, which you are studying from the class 10th, right? Now let's talk about here with respect to the transpiration and photosynthesis. A compromise means this is a very interesting topic given in your NCERT textbook. Okay, so see, transpiration has more than one purpose. Why the transpiration has to do? Because there is a lot of reasons are there. So जैसे कि see, It creates a transpiration pool for the absorption and transport of the plants. Understood? Next, supplies water for the photosynthesis, then transport minerals from the soil to the other parts of the plants, then cools the leaf surfaces, sometimes 10 to 15 degrees by the evaporative cooling technique. Okay, next, maintain the shape and the structure of the plant by keeping the cell turgid. Okay, so these are the plus point we have with respect to the transpiration as compared to the photosynthesis. Now see, an actively photosynthesizing plant has an insatiable need for the water. Photosynthesis is limited by available of the water, which can be swiftly depleted by the transpiration. Means what? When the water transpiration process is happening continuously, water will be getting lost. Okay, because of this, the photosynthesis process can be in a problematic condition. Okay, now see, humidity of the rainforest. Humidity of the rainforest is largely due to the vast cycling of the water from the root to the leaf to the atmospheric and the back to the soil. So this is how the cycle has been explained over here in very short paragraph. Okay, let's talk about here with respect to the C4 plants. The evolution of the C4 plant is probably one of the strategies for maximizing the availability of the CO2 so that they can fix the CO2 very proper way, okay, while minimizing the loss of water. ये उनको करना ही होता है. Okay, now see, C4 plants are twice as efficient as compared to the C3 plants in terms of fixing the carbon. Why we have to fix the carbon? Carboxylation process has to take place. Why? So that we can just make a particular sugar, that is a glucose, okay, that can be stored in the form of starch. Everything you know now very well. Okay, so however, a C4 plant loses only half as much water as C3 plants for the same amount of the CO2 fixed. मतलब जितना amount में CO2 fix होता है C3 plant में, okay, उतना ही अमाउंट में C4 प्लांट में भी फिक्स होता है, लेकिन जो वाटर लॉस होता है वो C3 प्लांट्स में ज्यादा होता है एज कंपेयर टू द C4 प्लांट्स में खाली आधा ही होता है एज कंपेयर टू C3 प्लांट्स. Okay, this is what the meaning of this statement. Now let's talk about the transpiration, a very important topic, you can say transpiration over here, that is the opening and the closing of the stomata. Okay, what is this topic called as the opening and closing of the stomata? Before this you should understand that it is called as a potassium ion theory. Okay, and here how the opening of the stomata will take place and how the closing of the stomata will take place, that is I am going to describe over here. But this concept is known as the potassium ion theory, which was given by the scientist called as a Levitt, very important person. Okay, now you know that during the daytime cell becomes the turgid and in the nighttime cell becomes the flaccid. Because of the turgidity stomata has to open and because of the flaccidity the stomata has to close. This is a normal basic concept which you are keep on studying, right? So now see, this is what structure I had drawn over here. This is a guard cells. I hope you know the stomata. Let me take you to the white screen first. Okay, so let's talk about here with respect to the stomata now. See, this is the guard cells, bean shape है ना. Okay, just wait. Let me clear this. Let me draw this proper way. Suppose this is a guard cell of the stomata. Okay, and these guard cells will be having the membrane also, but thicker at the center part. Thickness will be more over here as compared to the peripheral region. Understood? So here there is a thickness is more. Okay, you should keep this all thing in your mind very clearly. Other than this, these guard cells will be having some kind of different structures also, like there is a presence of large vacuoles over here. Okay, so this can be a vacuoles. Just listen to my statement, that is very important to you. Rest everything will be showing with respect to the diagram also. Okay, now here there will be chloroplast arranged. Okay, so this green color because of the chlorophyll. Okay, so here there will be the chloroplast present over here. Other than this, there is a subsidiary cells, even the nucleus will be present over here in the guard cells. Okay, and now there is a subsidiary cells from the surrounding. Okay, this is how the subsidiary cells look like. Fine, so this is how the stomata look like. And you know that in between this pore is that this is nothing but the stomatal pores, but the guard cells will be also having some strands over here. Okay, these are the tonofibrils which you should keep in the mind. Okay, so these are the fiber like substances which will be helping the guard cells to get open up during the turgidity. Okay, so that the water vapor will be going out, means, and the carbon dioxide will be getting inside. Okay, so CO2 has to move inside and the water vapor has to come outside. So this is a normal procedure which is happening during the opening of the stomata. And then then when these guard cells become flaccid, the stomata has to close. This is the normal procedure which you know very well. Okay, so let's move on to the further explanation. This is a dicot stomata, and suppose if I talk about the monocot stomata, what is the difference? It shows a dumbbell shape guard shape. Okay, guard cells are in a dumbbell shape. Okay, so can you see over here? So this is how it looks like, and the thickness will be more in the center part of the dumbbell shape. Okay, at this region there will be the more thickness. Rest everything will be remain normal, only a fine membrane like structure, and rest everything again the same. Like here there will be the subsidiary cells. Okay, rest all is the same thing. Activity is also performed same. Now after understanding these parts, let's go to the PDF now. How the stomata is getting open, that is a very important topic for you to understand. Focus over here very clearly, because the question can asked in the examination over here. So these surrounding cells which I had drawn over here, these are all subsidiary cells. Okay, consider as a subsidiary cells. Fine, labeling is done over here. Now as I told you, these guard cells will be having the vacuoles, having the chloroplast. Okay, this normally two things you'll be able to see. Also the guard cells having one more kind of channel like opening. Okay, this channel is called as a hydrogen potassium ion channel. Okay, what is this channel called? As a hydrogen potassium ion channel. I'm repeating again, the chloroplast guard cells, sorry, the stomata guard cells will be having the vacuole, the chloroplast, and one more opening which is normally called as a channel, hydrogen potassium channel. Okay, now in the vacuole, what is going to happen, that we have to know. But before this, suppose there is a chloroplast. In the light reaction, photosynthesis has been taken place. Photosynthesis product is the glucose synthesized. Glucose will soon get converted into PEP. You already studied respiration part, right? In that you know that C4 plant cycle, right? In that you studied that it is PEP when comes in contact with the carbon dioxide. PEP itself is a 3 carbon compound. Carbon dioxide is a 1 carbon compound. Both will be fused together to form a 4 carbon compound called as the oxaloacetic acid. Same thing was explained in the C4 cycle also, in presence of an enzyme called as a phosphoenol pyruvate, right? Carboxylase. Remember the procedure. Okay, just go through the respiration part also, you will understand very clearly C4 cycle. Okay, now this oxaloacetic acid will soon get converted into malic acid. Now as soon as the malic acid has been formed in the guard cells, what is going to happen over here, that malic acid will be split up into the hydrogen ions and the malate ions. Malate ions will not be stopped in the guard cell, it will just move into the vacuole and it will remain over there. Okay, I'm repeating again, malic acid will break down into the hydrogen ions and the malate ions. Malate ions will enter into the vacuole part. Okay, now what will happen to the hydrogen ions, this will be concentration will be keep on increasing into the guard cells, because this procedure is keep on happening, right? So in the guard cell there is a hydrogen ion concentration has been increased. So what we have to do, when the hydrogen ion concentration has to be increased, that has to be going out from this hydrogen channel. Are you understanding? It has to go into the subsidiary cells. So things has been moving from the higher concentration to the lower concentration, because the subsidiary cells is not going to have that hydrogen ions outside, right? As much as compared to the guard cells. So guard cells has a higher number of hydrogen ions that has to move from the hydrogen channel into the subsidiary cells. Now when the positive charge is moving outside, inner side there is a decrease of the positive charge may possible. So for maintaining the balance, what will happen, the potassium charge, okay, the potassium ions from this again the same channel, okay, will enter inside the guard cells for maintaining the things neutral, or you can say for the balancing activity. So potassium is getting more in number. Are you understanding? From the subsidiary cells, the potassium has to move through the hydrogen potassium channel inside the guard cells. Okay, now the same number of hydrogen which comes out, the same number of the potassium has to move inside. Now what will happen to this potassium? See what will happen to this potassium. Now there is a positive charge has been increased inside, so this has to become neutral now. Okay, because the concentration of the potassium ion is getting increased. Now what will happen, from the subsidiary cell a chloride ion will start moving inside. Okay, from the subsidiary cells a chloride ions will start moving inside the guard cells. Okay, so that charge will become neutral, that is the potassium and the chloride will be in the same number, so that things will be become neutral. But you should also remember one more thing, it is an active process. Opening of the stomata is an active process. How? Because see, when the hydrogen ions comes out, potassium ions go inside. During this period of time, 1 ATP is getting used up. I'm saying that one ATP is going to be used up. Therefore, it is a, what you can say, a active process. Once the potassium ion is inside, malate ion is there already in the vacuole. So also the potassium ion will move inside the vacuole. It will be forming a potassium malate, okay, inside the vacuole. I'm repeating again, malate ion and the potassium will react together to form a potassium malate inside the vacuole, and the same procedure is going to be keep on repeating, and the guard cell will become turgid, and the what will happen, it will be start opening the stomata. So what is happening over here, you should recollect all the points from here. See, increase the ionic concentration, that is the K plus and the Cl minus concentration is increased into the guard cells. Okay, because of this, osmotic pressure is also going to increase, due to which the water potential is going to decrease, because the solutes are, means, see, these are the particles na, like potassium ions and the chloride ions, ion concentration is getting increased, so potential, water potential in the guard cells is going to decrease. Okay, therefore the water has to move from the, see, water has to move inside the guard cells from the subsidiary cells, that is the surrounding cells. So are you understanding? So from the subsidiary cells, water will also move into the guard cells, because guard cell concentration means water potential has been decreased, and outer side the subsidiary cells has a water potential more in number, therefore the things has to move from the higher water potential to the lower water potential. Okay, very clearly understood from this. So now guard cells will, what will happen to the guard cell, it will become turgid and the stomata will be opening. Okay, and the stomata will be opening. If you don't understand this process very clearly, just move back again, try to listen, you will understand the concept very clearly. Okay, now going further with respect to the closing of the stomata over here. Now see, during the night time, no photosynthesis is possible, due to which the carbon dioxide concentration is going to increase. Okay, because in the night time, there is no light, so no photosynthesis. So carbon dioxide, which has to fix into the cycle, which is not possible. So his concentration is going to increase inside the guard cells. Okay, results in the acidic pH, because this you can say carbon dioxide is going to react with the water, and many things, many reactions will be taking place. Because of this, the acidic pH is going to increase in the guard cells, which results in the trigger of ABA, that is the abscisic acid, because it is in the stress condition. So ABA will inhibit the potassium ions process, means what, the normally potassium which has to move into the guard cells, and the ionic balance will be getting disturbed, then the water from the surrounding cells will enter inside the guard cells, and you know that it will become turgid and all those things. But this is not going to happen during the night time. Okay, because the potassium pump has been stopped because of the abscisic acid. And who triggers the abscisic acid to active, carbon dioxide concentration, right? Because of this, the cell will become flaccid and it will be like closed, stomata will be getting closed over here. Okay, cell has been become flaccid, guard cells has been become flaccid, and the stomata will be closed over here. So this is what the opening and the closing of the stomata has been over. Now, what are the things which can affect the transpiration over here? See, humidity. Higher the humidity, slower the transpiration. If lower the humidity, faster will be the transpiration. Coming to the light, okay, that is the second factor. Light intensity increases. Light increases in the photosynthesis, result in the glucose preparation. You know that during the photosynthesis, glucose preparation increases in the guard cells, which results in the hypertonic condition, due to which the ions from the subsidiary cells move into the guard cells and become turgid and open up, results in the transpiration. This is the same scenario which I had explained right now to you, right, into the guard cells. Because of this only the guard cell was getting open up, right? Temperature, higher the temperature, more transpiration. Stress hormone is getting activated if there is a temperature, more high temperature, and this ABA is going to close the stomata so that excess of transpiration will be avoided, so that plant can be like storing the water now. Okay, so this is the normal three factors will be seen over here. Now we are entering into the very new topic, that is called as the uptake of the minerals, that is happening. See, mineral absorption is mainly active or it is very less passive. Okay, now active absorption, if it is doing, there is a two reasons why it is required. First reason is that minerals are present as ions in the soil. What I told over here, see, minerals are present as ions in the soil, and then see, the cell membrane does not allow the entry of this charged particle inside, therefore the active absorption has to be taking place. Next, the concentration of the mineral ions in the soil is very very less. Again, you know that if the person is spending his energy, so concentration ke against काम करेंगे ना, that means from the lower concentration thing has to move through the higher concentration area, right? So now same thing, let's talk about here more in detail. First of all, there is a note. Note says that specific proteins in the membrane of the epidermal cells actively pumps the ions from the soil into the cytoplasm. Okay, actively means what? Expenditure of 1 ATP. So specific proteins can be present on the membrane epidermal cells, which will be like pulling the ions from the soil inside the cytoplasm with the help of 1 ATP. Okay, therefore the active transport. So transport protein of the endodermal cells are the checkpoints. Means endodermal, you know very well where it is present, right? So they are the checkpoints where the quantity, okay, quantity and the type of the solute that reaches to the xylem is has to be checked. Means what kind of solute has been coming over there in the xylem, it will be checked up. Okay, the cells of the root endodermis have the ability to actively transporting the ions only in the one direction. So obviously from the root as the things has to move to the towards the xylem, it is showing a single direction, that is from the root as to the xylem part, right? It is a single direction. Now next point is that see transpiration only, lekin we are going to deal with respect to the new topic, that is translocation of minerals. Okay, how minerals are getting translocated over here. Minerals move along with the water. Okay, minerals always move along with the water. The sink for the minerals are, see, sink means what? Where these minerals has to be getting stored, or where the part where it has to be transferred. Okay, so mineral meristem is the one region, younger leaves is the one region, developing seeds are the region, then the fruits and the storage organs where these minerals can go and work accordingly, or they can be stored accordingly. Understood? So these areas will be called as a sink. Very very important. Okay, because sink theory we are going to study, sink theory we are going to study in detail just after this. Okay, so unloading of the mineral ion takes place at fine vein ending by the diffusion or by active uptake. Elements like calcium, okay, which are a structural component, cannot be remobilized. You know that calcium is not going to move, so it has to require some kind of carrier proteins, then only the calcium can be uptake. We will be studying in the coming chapter, that is the mineral nutrition. Okay, in that part we will be dealing about some kind of elements, okay, mobilized element and immobilized element, that is going to be explained. So don't worry about that calcium part. Let's move ahead. Analysis of the xylem. Okay, exudates shows that most of the nitrogen is carried as an organic compound. Very very important statement, straightforward questions can be coming from here. These are ncrt lines. Okay, analysis of the xylem exudate shows that most of the nitrogen is carried as an organic compound, that is in the form of amino acids, and very less as a inorganic ions. Okay, तो ज्यादा करके कंपाउंड जो है वो ऑर्गेनिक कंपाउंड्स में लिए जाते हैं एज कंपेयर टू द इनऑर्गेनिक आयंस, जो कि बहुत कम में लिए जाते हैं. Small amount of the phosphorus and sulfur are always carried out as a organic compound. Very very important part. Again, keep this in mind. Phosphorus and sulfur are taken as a organic compound. Now this is our last part. So let's try to go with this point. Very very important part of the lecture, you can say. See, translocation of the food through phloem. Okay, now we are talking here about with respect to the food, and the yes conducting element has been changed over here. Now it is a phloem. Everyone knows that food has been conducted with the help of the phloem only. Okay, so phloem shows the sieve tubes, sieve cells, and the companion cells, which we had already studied in the anatomy of the flowering plant, right? Sieve tubes always be there with respect to the companion cells, and the sieve cells will be always be there with respect to the albuminous cells. Okay, sieve tubes and the sieve cells which are going to be matured, and they will be like enucleated, whereas companion cells and the albuminous cells was the nucleated cells. If you don't remember, just go through the anatomical lecture video, you will understand, okay, very clearly. Now try to understand this concept. Movement of the food is a mass flow hypothesis. Okay, it is called as a mass flow hypothesis, and this theory is also called as a source to sink theory. Source means where it has been produced. See, source means what? Food is produced over here. It can be the leaf region where the photosynthesis is actually happening, and sink is where it has to send. Okay, or where it has to be stored. That can be leaf also. That can be the storage areas also. Storage area, like suppose if I take example as a sugar cane, so interzonal region, internodal region is the responsible for the storing the sugars and all. Okay, like this way you will understand many things over there. But here we are talking about with respect to the source to sink theory. Means Now source, you know already how the photosynthesis already takes place, how the food has been prepared, and all those things you very well know. So there are some points which you should keep in the mind, like see, carbohydrate synthesis as a glucose, transported as a sucrose, and stored in the form of the starch in the plants. Okay, note, transport of the food is a multi-directional process, because food has to be supplied upside also, downside also, side by side also. So everywhere there is a requirement of the food, and it has to be sent through phloem only. Therefore it is a multi-directional process, whereas xylem movement is only the unidirectional process. Okay, so you should able to differentiate between the xylem and the phloem in this way also. Let's talk about here. See this elongated structure which you see on the screen, this is a normal cu tubes. Okay, and here there is a companion cell, you can see a dot like structure inside, that is what the nucleus. Okay, that is what the nucleus of the companion cells. Understood this much very clearly? Now what is happening over here? Now suppose the food has been prepared, and that food has to be poured inside the phloem. Okay, pour inside the phloem. When the phloem pours the means see, when the food has to be transported, always it is transported in the form of sucrose, as I told you now. Okay, it has been transported in the form of sucrose, so it will be not directly added into the cu tubes, it will be going through the companion cell, because they are only living. Understood? So now the companion cells के through sucrose अंदर जाएगा cu tubes के अंदर. So you come to know that sucrose concentration is going to increase inside the cu tubes. That is nothing but a hypertonic condition. Are you understanding what condition has been going to be seen under the cu tubes, means inside the cu tubes? That is a hypertonic condition. Sucrose concentration has to increase. So you know that when the hypertonic condition is there, and the neighbor cells, whatever the neighbor cells will be present over here, these all neighbor cells will be having water content in that. That is a hypertonic condition. So water will be always move from the hypertonic condition to the hypertonic condition, है ना? So when waters from the neighbor cell when enter, see what is written over here, water from the surrounding cells moves in, that is inside the cu tubes itself. So what are the two things are there in the cu tubes? Now there is a presence of the sucrose, and now there is a presence of the water itself. So now the pressure will be started build up. Okay, now the pressure will be more inside the cu tubes. Now because of this pressure, things will be moving from the high pressure to the low pressure area. Okay, so just below that can be the another cu tube present over there. Okay, so things will be started moving downside. Suppose there is a presence of the cu tube on the upper side, so things will be moving to the upper side, right, into the low concentrated area. Understood? So this is how the things will be moving from the high concentration to the low concentration. Suppose it has to move to the side by side also. Okay, so it will enter into the side by side also. Now suppose it has to go and store, it has to be stored. So while storing, see, from the sieve tubes, the things when enter into the storage organ, okay, it can be anything, it will be always stored in the form of the starch, but this will be an active process, means it is an ATP required process. Okay, what is this process called as a ATP required process? It is an active process. So whenever the sucrose has to be stored into the storage organ, it is an active process. Okay, keep this in mind, very very important and easy concept over here. Just try to go back and try to analyze, try to read this again and try to understand again. You will understand the concept over here. If it is not understood very clearly, just let me know, I will be clearing your doubts again. Okay, now we are completed the chapter called as a transport in the plants, but before completing, there are some important points which can be asked in the examination, so you should familiar with that. No root pressure in gymnosperm will be seen. Imbibition pressure in the P is almost approximately 1000 atmospheric pressure. Cohesive force is nothing but called as a tensile strength of the water. You just read NCERT, you will come to know all these things. Okay. Then psychrometer, it is an instrument which is a helps in measurement of the relative humidity and the transpiration. Hydrometer is used for the measuring density, or you can say gravity of a liquid. Same thing, tensiometer, measuring the soil water tension. Okay, what is the purpose? Measuring the soil water tension. Mammometer is there, measuring the root pressure. Barometer is there, measuring the atmospheric pressure. Barograph is there, that is nothing but the recording will be seen on the barometers. Okay, next, porometer is there, which is helping measuring of the size of the stomata. See, atmometer is there, which is helping in measuring the pull caused by the water evaporation from the porous spot. Okay, from the porous spot. Okay, keep this in mind. And the potometer is there, which is helping in the measuring the transpiration pull. Potometer is for the transpiration pull. Okay, keep this in mind. Next, cryoscopic osmometer is there, so measures the osmotic potential of a solution by measuring its freezing point. Hollard means what? So total amount of the water present into the soil, that is Cressard plus Eckhart. So what is Cressard now? Available of water for the plants, that is the capillary water. Okay, that is considered as a Cressard. And Eckhart means what? So non-available water, that is combined water as the water of crystallization, or you can say hygroscopic water, as absorbed, adds adsorb water on the soil particles, and the gravitational water which is downside, you can say. So that is not easily available to the plant. Such water will be called as a Eckhart. Okay, and the water which is very easily available to the plant, it is called as a Cressard. Okay, and Hollard means combination of this Cressard and Eckhart. So here we had completed an interesting chapter called as a transport in plants. Go through it. Okay, you will understand very very easily, and read your NCERT textbook compulsory. Okay, मिलेंगे आपको नेक्स्ट लेक्चर से.